Do you know what is Protein Data Bank and what it is used for? You might have heard a lot about PDB in bioinformatics, in drug discovery and solution, but don't know what it is. It is still confusing how it has been used, what is the application, how it can be applied to drug discovery and finding its solution. So various protein receptor molecules, how people are using, how researchers are using, you're still confused. Papers are making you confused. There are so many scientific literatures which are known, but you are not able to understand the materials and methods which they are following. Right? So here in this video, I will show you step-by-step process of how PDB is used and how it can be applied and what is the history and everything other information that will make benefit to you. The Protein Data Bank, which is also known as PDB, is a global archive of structural data for biological macromolecules. PDB was established in 1971 at Brookhaven National Laboratories. The goal of PDB includes making the archive as consistent and error-free as possible, ensures that the final archive is kept uniform. Now, who runs it? The data is freely accessible on the internet via the websites of its member organizations such as Protein Data Bank in Europe, Protein Data Bank Japan, Biological Magnetic Resonance Data Bank, and Electron Microscopy Data Bank. Now let us understand how PDB can be utilized in drug discovery and solution. So PDB is a database for 3D structural data of large biological molecules such as proteins and nucleic acids. So the data typically obtained by X-ray crystallography, NMR spectroscopy, or cryo-electron microscopy. So PDB is overseen by an organization called the Worldwide Protein Data Bank, that is WWPDB. Each new structure is deposited in PDB, which is currently run by Research Collaboratory in Structural Biology, that is RCSB. The structures can be downloaded from RCSB's PDB web server, which also provides additional information about each one. Further information, some of it focusing on specific types of molecules or specific aspects of the molecules, can be obtained from a large number of other structural databases on the web. Now let us understand how this works. So you can go to search and there are so many search options. So basically you can go to any kind of option. So I'm just clicking on the first option. So when this page opens, you can see there is an option over here that is to mention this search term or entry ID or sequence number. So here you can add any kind of protein. Say I am searching for keratin, so there are so many options for keratin as well. So you can select either one of this, and here you get so many options. So generally, PDB ID is 4-character unique identifier of every entry in PDB. A 4-character PDB ID is assigned to each new structure at the time of deposition. The first character is numeral in the range of 1 to 9, while the last 3 characters can be either numerals, that is in the range of 0 to 9, or letters in the range of A to Z. So if the PDB ID of an entry in PDB is known, it is the most direct way to retrieve it from the database. The PDB file for a protein contains detailed information about the protein, including the position of each atom in 3-dimensional space. The design of this PDB web page allows easy navigation and provides different options to find the structure of interest, facilitate in finding similar structures, or jump to related contents in different databases. So this page is organized into sections that includes different types of informations. It shows the accession code and the information about the authors, the deposition date. There are many such informations related to this protein PDB ID. So this section of macromolecules shows the entry ID about the chains, the sequence length, the organism from where it has been taken, also the biological assembly as previously shown in the top. then it shows various other small molecules which is linked to this protein, such as in this one there are two ligands attached with different kind of chains. The name of this ligands, the chemical formula, all these are mentioned over here. Experimental data snapshot. This section is devoted to the X-ray crystallography experiment and the statistical data revealing the resolution of the structure. In case of this PDB ID, the structure was determined at resolution of this much Armstrong, which is not a very good resolution. Resolution refers to the quality of the experimental data generated by X-ray crystallography. High resolution structures will be determined at a values of less than 1.5 Armstrong or so. This level of accuracy of determining the atomic positions is high. Conversely, at resolution of 3 Armstrong or higher, the structure shape as a global will be inferred. So, however, the accurate positioning of the individual atoms is poor. Further information, some of it focusing on specific types of molecules or specific aspects of the molecules, can be obtained from a large number of other structural databases on the web. One such database is PDBsum. So for this, first of all, we will just copy paste the PDB ID, that is the 4-digit PDB ID from our Protein DataBank website, and from here we copy and paste the 4 characters, and after that we click on Find. So PDBsum is web-based database providing a large pictorial summary of key information on each macromolecular structure deposited at PDB. It includes images of structure, annotated plots of each protein chain's secondary structure, detailed structural analysis generated by ProMotif program, summary project results, and schematic diagrams of protein ligand and protein DNA interactions. In the list of molecules that follows, each protein chain is shown schematically by a wiring diagram depicting its secondary structural motifs, primary sequences, structural domains, and highlighting the active site residues, the residues that interact with ligands, metal or DNA, RNA molecules. The secondary structural motifs are computed by ProMotif program, whose detailed outputs are available via hyperlinks, while the domain definitions come from CATH, Protein Structural Classification Database. For each ligand molecule, a lick plot diagram gives a schematic depiction of the hydrogen bonds and non-bonded interactions between it and the residues of the protein with which it interacts. The right corner contains a hyperlink to download structural files of this accession code for the PDB file extension, in addition to other types such as PDBx, CIF files. So the typical form of storing 3D structure information is PDB file format. And these are some of the references that I wanted to share with you guys. These references are really helpful. Please go through the references which are also listed in this paper. They are majorly about PDB database and PDBsum, how to use it. These are somewhat old. You can go through some of the new literatures as well. I hope from this video I have illustrated most of the things which are required to understand PDB and which is required to use it. This video is particularly for them who are new to bioinformatics and they are struggling to understand how computational approaches can be utilized. Thanks for watching, and I will see you in the next video.